Hi everyone, let us understand about restriction modification system which is present in bacteria. This restriction modification system can be compared with the human immune system. So when a foreign organism enters into body, host organism, so the immune system of the host organism detects or identifies or recognizes the foreign organism, and it kills the foreign organism, or it removes the foreign organism from the body. Similarly, this restriction modification system is a type of immune system present in the bacteria. This restriction modification system, whenever a foreign organism enters into bacteria, so that foreign organism is uh, killed by this particular restriction modification system. How many types of restriction modification systems are present in bacteria? Four types of restriction modification systems are present in the bacteria. So this can be compared with the types of immune system, types of immune system in humans. For example, we have acquired immune system, and also we have natural immune system. So innate immunity is there, and also the acquired immunity is there. So we can compare these four with that innate immunity and acquired immunity. That means their action is specific, because you know that innate immunity and that acquired immunity, their action is very specific. And similarly, this type 1, type 2, type 3, and type 4, so the process of eliminating this foreign organism by this type 1, type 2, and type 3, and type 4 is different. Okay. uh, this restriction system, let's understand about the restriction this restriction system. As I said, this removes these foreign organisms from the host organism, that's a bacteria. And we know that what are the foreign organisms that can attack the bacteria. So in case of humans, we know that there's different types of uh, pathogens, or different types of parasites, and different types of organisms, viruses, and so such things may enter into human body. Whereas in bacteria, only viruses can enter, not any other types of organisms can enter into the bacteria. So bacteria is susceptible for the attack of the viruses. So viruses that can attack the bacteria are called bacteriophages. Okay. So that means this restriction modification system, it clears or removes the bacteriophages from the um, host, that is the bacteria. Okay. Coming to this restriction modification system, so this is a enzymatic system. So in a humans, there we know that different cells are present and different proteins are present, which participates in the immune reaction. Similarly, in bacteria, here cells, because they are unicellular organisms, so we don't find the cells to function against these viruses or bacteriophages. Okay, we could expect only the proteins. Okay, the proteins can they can involve in this particular type of immune function. So the proteins that involve in this function are called restriction endonucleases. Restriction endonucleases, the term itself indicates that. So endonucleases means these proteins, they act on the nucleic acid, or the DNA, or the genetic material of the viruses. So since we said they're endonucleases, they can cut the DNA. They cut the DNA not at the ends, so they cut the DNA in the middle of the DNA. So coming to the structure of this proteins, see, as we know that four types of systems are there. So all these four systems, they function through the in proteins only. So these proteins are called endonucleases. But all these four may exist in the same bacteria, but their action is different. But all they function through the proteins only. Okay, coming to the type 1 restriction system. So this type 1 restriction system has a protein. This protein is made up of 3 subunits. Whereas type 2 system, this type 2 system has only 2 such units are present in the type 2 system. Okay. When we see the type 3 system, there we can see that no, this also has two subunits. So type 4 also has the two subunits. Only type 1 has the three subunits. So the difference is that no, type 2 and type 3 are type 4. So type 2 and type 3 type 4 are different. They are not different subunits. They are completely different proteins. Okay, whereas the type 1 is a different subunits are there in the type 1. Okay, so you know that any enzyme to function in a cell, it needs some cofactors. So the cofactors may be like some ions which are present in the cytoplasm. So the cofactors which are required for the function of these enzymes are magnesium ions in case of these endonucleases. Okay, and why they are named as a restriction endonucleases? So because they restrict the propagation of the viral DNA instead of the bacterial. Therefore, they are named as a restriction endonucleases. So we are going to understand how these Mg plus ions are important for the function of this endonucleases. We know that the same Mg plus ions are required for formation of the phosphodiester bond during the synthesis. That means even the polymerases, they use Mg plus ions for their function. At the same time, to break the phosphodiester bond also between the nucleotides in the DNA. So the same magnesium ions they play a role. Okay, so coming to this restriction modification system, as I said, no, this recognizes the DNA. So recognition of the DNA when two genetic materials are present in the cell. Suppose the bacterial DNA and the viral DNA both are present. So for these enzymes, identifying which is the bacterial DNA and which is the viral DNA becomes a challenge. So these enzymes have a function that, so these enzymes easily can differentiate which is the bacterial DNA and which is the viral DNA. So what are the specific features that makes these enzymes to recognize or to differentiate the viral DNA from the bacterial DNA? So bacterial DNA during the replication. So during the replication, when the new daughter DNA strand is synthesized, so the parental DNA strands usually they are methylated by the methyl transferases. So even during replication, when a daughter DNA strand forms from the parental DNA strand, so even for repair mechanism, we know that so that repair enzymes, they differentiate the daughter DNA from the parental DNA, because the repair enzymes they don't make any edits on the parental DNA, they only make edits in a daughter DNA. Even to differentiate the daughter DNA from the parental DNA during the replication, so for proofreading activity, so they rely on the methylation of the DNA strands. So parental strands are methylated. So the daughter strand is not methylated, parental strand is methylated, and daughter strand is not methylated. Therefore, we see the DNA in the hemimethylated form. So the repair enzymes, they start making changes in the daughter DNA. The same methylation is used by this restriction endonucleases to differentiate this viral DNA from the bacterial DNA. So when viral DNA enters inside of the cell, so the viral DNA is not methylated, whereas the host DNA is methylated by the methylases. Therefore, this restriction modification system, it initially recognizes, or sorry, initially it recognizes this viral DNA from the bacterial DNA. Once the viral DNA is recognized, and once the viral DNA is recognized by this restriction modification system, okay, then this same protein, the same protein which has that restriction function, it identifies a specific sequence on the viral DNA. So though the viral DNA is recognized by this RMS system, the RMS system immediately upon identification of this viral DNA cannot make cuts on its cannot make a nicks in a DNA of the virus. So it search for, or it scans for a specific sequence on the DNA. So the sequences which are recognized by these particular proteins, this restriction modification system proteins that we call endonucleases, okay, these specific sequences are called the restriction sequences. Okay, we call the restriction sequences because these sequences, presence of these sequences initiates the endonuclease function of this restriction modification system. Okay, once these sequences are identified on viral genome, viral DNA, so then the endonuclease function begin. Okay, so when the endonuclease function begins, so the function of this endonuclease in a different this RMS systems is different. That means in the type 1 RMS, type 2 RMS, type 3 and type 4, so their recognition sequences is different. That means the sequence they search for, they search on a viral genome is different. One, their recognition pattern is different, and also um, the recognition pattern is same. That means all they look for whether the DNA is methylated or not. So when they scan the viral genome, so the sequences they search is different, not the same. Okay, so type 1 they search a different sequence, type 2 they search a different sequence, type 3 they search different sequence, type 4 they search a different sequence, like that. Okay, type 1, when we see this is called as a bipartile recognition sequence, we call it. So when we say restriction sequence, not, you should not say it's a recognition sequence, you can say it's a restriction sequence. So bipartile sequence means this restriction sequence is separated by a segment of DNA. That means we see two parts in a sequence. So these sequences are separated by again a small sequence which is not important for its endonuclease function activity to be initiated, for initiation of the endonuclease activity. Okay, the type 2 where it's not a bipartile. Okay, and type 3 it's not also bipartile, but when we see type 4, it's a bipartile sequence. And symmetric and asymmetric conditions. So when we say symmetric condition, so suppose 6 nucleotide sequences are there. Suppose when you draw a vertical line, okay, between the third and fourth bases, so when we look on the left and right sides of the sequence, so the sequence of the left side matches with the right side, or I can say the left side sequences is a mirror image of the right side sequence. So such sequence is called a symmetric sequence. So that symmetry of the sequence is seen only in the type 2. That means only type 2 restriction modification system it search for such symmetric sequence. Okay, whereas type 1 it doesn't care about the such a symmetric sequence, so it searches for the asymmetric sequence. Though it's the asymmetric sequence, it's not only the asymmetric sequence, it's a bipartile sequence that we should understand. The fourth one is a bipartile sequence that we have to understand, and the third one is also asymmetric sequence. It's not a bipartile, but it's still is the asymmetric sequence. Okay, so next important thing is that that we have to understand is the methylation. So coming to methylation, this restriction modification system itself has a methylation functions. So cannot it has a subunits that can methylates the DNA. So for example, adenosine methyl transferases so is there. So this adenosine methyl transferase it methylates the bacterial DNA. So type 1 and type 2 and type 3. So in type 1, so they completely they have this kind of that means function, that a restriction endonuclease itself has such function. Type 2 RMS system has such function. Type 3 RMS system has such function. But type 4 doesn't have such function. So that you have to remember. Okay. So this is the illustration which explains how that RMS system removes the genome of the virus when this virus enters, when virus injects or inserts its DNA into the cytoplasm of the bacteria. Thus the cytoplasm of the bacteria has this RMS system. So this RMS system, as I said, no, it has adenosine methylase activity. This adenosine methylase activity, it methylates the host DNA. But when a foreign DNA enters inside, genome of the virus enters inside of the cell, so this genome doesn't have any methyl groups. Therefore this restriction system recognizes recognize this foreign DNA, DNA as a foreign DNA, and then its endonuclease activity. So the same system search for these sequences, bipartite sequences and symmetric sequences and asymmetric sequences, and then the endonuclease breaks down the phosphodiester bonds which are present between the nucleotides. So that is how this restriction modification system works. Now let us understand how these magnesium ions they what is the role of their these magnesium ions, how they function in breaking of this phosphodiester bond. So magnesium ions, we know that these same magnesium ions are important for the formation of the phosphodiester bond. So they work along with the DNA polymerase enzymes. Similarly, the same magnesium ions now they work with the endonucleases. Okay, so the amino acids like aspartate and glutamate, which are present in the this endonuclease protein. So these aspartate and glutamates they keep these magnesium ions in the active site of these endonucleases. So these are cofactors, and these cofactors they bind with the aspartate and glutamate. Okay, so this divalent metal ions, suppose this metal magnesium ions, so they have positive charge. Okay, now we know that there is a phosphodiester bond is present between the bases, like here we can see this OPO is a phosphodiester bond. Okay, now this magnesium ion, so two magnesium ions they participate in this particular process. So in even the in DNA polymerase they utilize the two magnesium ions. We know that why they use one magnesium ion for the nucleophilic attack, so another one to neutralize the charge on the phosphate group. Similarly here one magnesium ion, so it starts it forms a bond with the oxygen which is present in the OPO phosphodiester bond. So the charge on the oxygen is neutralized by the magnesium ion. So when the oxygen the charge on the oxygen is neutralized by the magnesium ion, now the oxygen group which is present in the water molecule, it starts forming a bond with the phosphate now. So the first oxygen, the charge on the oxygen is neutralized by the magnesium. Then the oxygen which is present in the water molecule, it starts forming the bond with the phosphate group in the OPO bond, phosphodiester bond. Therefore, so the bond between the oxygen and phosphate in the OPO it breaks down. Okay. Now the second magnesium ion. So this second magnesium ion. Now the second magnesium ion it neutralizes the charge on the other oxygen atom present which is present to phosphate. Okay, now the OH group which is present in the water molecule, it will form a bond with the phosphate. So this is how this magnesium ions and oxygen with the help of the oxygen they make a break between the bases. They cut the phosphodiester bond.